Okay, thank you, Chairman, and also thank you to the organising committee for inviting me here. It's a great honour. I'm very pleased to be here, enjoying it very much. So what I want to do today is to talk about our work on rice photoprotection, which is something we've been doing for about the last 15 years now. And this follows on from talks earlier in the week on photosynthesis. So there was a lot of discussion about C4 photosynthesis and photorespiration. So what I've done on this slide here is to list other aspects of photosynthesis that really could be improved. And this is from a review by Matthew Reynolds, but it applies to rice as well. And we can see, for example, that we can improve the absorption of radiation by canopies, but we expect maybe only a few percent, as was mentioned by Jingguang Zhu yesterday. We could raise the leaf photosynthetic capacity, that's the light saturated rate of photosynthesis, via different mechanisms, and we would expect something like 10% improvement depending on conditions, canopy structure, the proportion of the canopy that was actually saturated by light. And of course, all of the, see if my pointer works, all of these C4 and Rubisco-related mechanisms will give us a large increase in photosynthetic capacity of our crops. There's one right at the bottom there that I want to draw your attention to, and that is it's not discussed much, it's the optimization of the distribution of photosynthetic components, or the photosynthetic properties of the canopy. So, for example, we know from modelling work that we could get up to 30% increase in canopy photosynthesis based on one aspect, and that's the slow relaxation of the quantum yield of CO2 assimilation in fluctuating light. So if I just follow that up, this is a sort of summary of canopy processes, and as we go from the top to the bottom of the canopy, we can pick out processes which result in a progressive lowering of photosynthetic efficiency. So, for example, at the top here we have losses according to non-PAR radiation absorption, reflection, saturation I've mentioned, and what I want to talk to you today is photoinhibition and photoprotection of photosynthesis in the canopy. And then lower down here you have other aspects like respiration and light harvesting. So the goal here is an integration of photosynthetic properties with the physical canopy components. Okay. So you probably sit there wondering, what on earth is he talking about? What's photoprotection? Well, I'm going to explain what photoprotection is and also how it's related to abiotic stress. I'll talk a little bit about some transformation work we've done over the last 5 years or so, and I'll talk about the work we've started with mutant screening, and lastly an integrated strategy, or what I think is an integrated strategy for improving photoprotection, which is something I think is actually extremely important and rather overlooked. So I always forget to I don't leave enough time for acknowledgements at the end, so I just wanted to point out that the photoprotection work in my lab is carried out by a set of people, mostly Stella Hubbard, Liang Zhao, Buki Ajikboy, and Ian Smiley, and also Dr. Xiaobing Peng and Sasha Rubin are very important. I'm going to discuss some mutant screening work at Erie, so a shout goes out to the team at Erie, the C4 team at Erie. So, as a leaf absorbs light, as chlorophyll absorbs light, it photosynthesizes, and as chlorophyll absorbs light, it's excited by that light. Electrons go into a higher energy state. But the problem is that photosynthesis saturates at a fairly low light level, and shown here. And this means there is an excess amount of excitation energy within the pigment bed of the thylakoid membrane. And as the light goes higher, the amount of excess excitation energy increases, and this is potentially actually dangerous, because if these excited states of chlorophyll live for long enough, they can react with unwanted things and cause photooxidative stress. And at very high light, we can sometimes see a decline in the rate of photosynthesis. And this is referred to as photoinhibited photosynthesis. So photoinhibition is the light-dependent decline in photosynthesis. It's worth pointing out that any factor, abiotic or biotic stress or nutrients or water, will lower the light-saturated capacity of photosynthesis and will increase the amount of excess excitation energy in the pigment bed. So even more dangerous. So what's the effect of photoinhibition on photosynthesis? Well, I've drawn these three lines here. If we plot photosynthesis against light intensity, the ideal line is shown by number 1 here. Okay, a photoinhibited leaf that's moderately photoinhibited will see a reduction in the quantum yield. Okay, a severely inhibited leaf will see a reduction in both the light-saturated capacity and the quantum yield. Okay, so this is always assumed to be damage to the photosystem too. It's not always the case. It can be simply quenching of chlorophyll that's sustained. So photoprotection. What's photoprotection? Well, as a leaf goes from light limitation towards light saturation, a series of mechanisms are kicked into place. And I'm going to refer to these biochemical mechanisms, but there are others as well. And what these do is they act in a photoprotective manner, and they prevent, or they help to prevent, this photoinhibited state from arising in the first place. Okay, and to an extent it's inevitable. You will always get photoinhibition, but the existence of photoprotective processes reduces that. So photoprotection actually refers to a huge range of processes and things that go on in the plant, both at a gross scale, so a morphological scale, and a biochemical scale. So, for example, if you take leaf morphology, we showed some years ago now that the upright leaves of rice plants, many rice canopies have erect leaves, and this acts as a very effective photoprotective mechanism from the bright light at midday. So light, instead of being absorbed by the leaf, is reflected down the leaf into the middle of the canopy. And this doesn't just increase canopy photosynthesis, it also acts very effectively to mitigate photoinhibition. And if you look at the chloroplasts themselves, this is a picture from a paper in Nature, but the chloroplasts actually move. And I think it's recently been shown that they also move in rice. This is Arabidopsis, but they move out of the way of bright light, and they can line themselves up along the inside walls, the vertical walls of plant cells, to avoid bright light. You get acclimation of photosynthesis, and you get something called feedback de-excitation, usually called non-photochemical quenching, or NPQ. And this is a down-regulation of excitation energy within the thylakoid membrane. And you get changes in electron sink capacity, and this can be almost anything actually. For example, photorespiration also acts to an extent as an electron sink. So I refer to all of these things as prevention of photoinhibition and prevention of photooxidative stress. And these two down here, damage repair rate and radical scavenging, are very important. Particularly the rate of repair of photosystem 2 is thought to be critical in determining the amount of photoinhibition. So many years ago we did some work at Erik in the dry season, and we worked out that there was substantial amounts of not just photoinhibition but also photoprotection going on in rice in these leaves. So if you look at this figure here, it shows light intensity throughout the day on a typical bright day in the dry season at Erik. And we get maximum levels of light. You won't see much higher light intensities than these tropical levels, about 2,000 micromoles per meter squared per second for most of the day, assuming no cloud cover. So what does that mean to photosynthesis? Well, if we plot photosynthesis against light intensity here, sorry, this should say photosynthetic rate, it actually saturates at just over 1,000 micromoles per meter, micromoles per meter squared per second. And this means that much of the light absorbed by the leaf in the field, in otherwise optimal conditions, is actually in excess of requirements. It's absorbing too much light, more than it needs. And this is associated with photoinhibition. So we measured photoinhibition using a technique called chlorophyll fluorescence, a very commonly used technique now, which measures the emission of light from chlorophyll. And the emission of light from chlorophyll competes with photosynthesis itself and a number of other processes. So by measuring the efficiency of the re-emission of light from chlorophyll, this is a reflection of the efficiency of photosynthesis. So we measure this parameter called FV over FM. It's a dark adapted value which gives us a measurement of photoinhibition, and we found quite low levels, dependent on variety, sometimes below 0.6, which is really quite a low level for FV to FM, and this is sustained for several hours. We also saw changes in the deepoxidation state of the xanthophyll cycle, which I'll explain later, which is a reflection of the extent of photoprotection that's occurring in the membrane. So what we got is this. What we would expect if we plotted light intensity against photosynthesis for a leaf, an unstressed leaf in the field, we would expect photosynthesis to move up and down these points between 1 and 2 fairly efficiently. When we do spot measurements of photosynthesis in the canopy in the field, we actually find that a lot of these points lie below this line, which came as a great surprise to us. For a crop plant in optimal conditions, you would expect them to be closer to the optimal response. You might expect this in a natural system with multiple stress, but we didn't really expect it in a rice crop growing at Erik. So we knew this was associated with photoinhibition. Also, we know it's associated with the deactivation of rubisco and also the accumulation of substantial amounts of carbohydrate in the leaf. So we identified what I'd like to call multiple failures of photosynthesis. So first of all, a decline in the light-saturated rate of photosynthesis, the onset of photoinhibition. This figure here shows a midday depression of photosynthesis. Okay, and we know that in the afternoon, for example, this decline in photosynthesis is associated with a decline in rubisco activity. We know from doing ACI responses at different times of the day, and this accumulation of carbohydrate as well. Okay, so we're building up a picture here of what's happening in the leaf under high light and high temperature. So we were interested in photoprotection at the thylakoid membrane level and identifying gene targets that we could use to manipulate the level of photoprotection, maybe enhance it. So these are mesophyll cells, this is a chloroplast, this is a thylakoid granule stack where the electron transport components and chlorophyll containing components exist. I'll just go through this very briefly for you. This is the light harvesting complex of photosystem 2. Photosystem 2 takes very basically takes electrons from water, passes them to photosystem 1 down an electron transport chain, and eventually these electrons will end up in the Calvin-Benson cycle or photorespiration. And we also get by the way, those two things also act in a photoprotective manner. By increasing the capacity for any electron sink in potential, you're increasing the capacity for photoprotection. The water-water cycle operates as an electron sink in a photoprotective manner, as does a cyclic electron flow around photosystem 1. But what I want to talk about is the light harvesting complex. Okay, and this absorbs a substantial amount of the light that's absorbed by chlorophyll within the leaf, and the excitation energy within the pigment bed of the light harvesting complex can be substantial. And there is a mechanism which de-excites the light harvesting complex. This is referred to as non-photochemical quenching, and this is a very important mechanism. And there are two components which regulate it. The first is a protein called PSBS, which sets the capacity for the quenching of excitation in the light harvesting complex. The second is the synthesis of the carotenoid zeaxanthin in high light, and then its reconversion back to violazanthin in low light, which seems to act in a regulatory manner. Okay, so we had some targets we wanted to look at in more detail, but the purpose of this talk is really to look also at the canopy level and look at biomass productivity. And I've been very concerned for a long time that there are trade-offs with processes like photoprotection. So, for example, photoinhibition and oxidative stress obviously will result in a loss in CO2 assimilation. I don't think there's any doubt about that, through a lowering of quantum yield and the convexity of the light response. And the thylakoid photoprotective processes help to prevent photoinhibition. But these processes come and go at various rates, and they will also result in a lowering of the quantum yield of photosynthesis. There's a possibility that there is a trade-off required between the amount of photoprotection that exists and the productivity of the leaf. Okay, so the question is, how protective is photoprotection, and is it ever wasteful? So, we wanted to do some genetics, and we started off with some transformation work and looking at its effects on biomass. We know photoprotection is quite a complex trait, therefore, and linked to many fundamental processes. So we questioned what genes we can target, but also what combinations of genes do we require to have an impact at the canopy level and under field conditions. So we started off several years ago by doing a microarray experiment, simply transferring leaves from low light to high light, and looking for responsive genes and highly expressed genes within the rice genome that seemed to respond to high light and to high light stress. Okay, and we were quite delighted that one of the most responsive genes is our friend PSBS here. Okay, there are also others such as beta-carotene hydroxylase, which sets the pool size of the xanthophyll cycle, which also regulates NPQ. There are also other stress-related genes such as superoxide dismutase and others. So we looked at PSBS in more detail. We felt this was a regulatory gene that really could have give us a good chance at controlling NPQ and controlling photoprotection. And it was discovered over 10 years ago now by Chris Nyoji in Arabidopsis, carrying out a screen of mutants that did not respond to highlight, looking for mutants that did not respond to highlight. And you can see here in this figure that if you overexpress PSBS, you get an increase in this non-photochemical, this protective non-photochemical quenching. And if you knock out PSBS, you still get a little bit, but effectively you got very low levels. So there's absolutely no doubt that PSBS determines the capacity of protective NPQ, and there's no doubt that this mitigates levels of photoinhibition in the leaf. There's also some nice experiments carried out in Stefan Janssen's lab, again in Arabidopsis, which show that when you grow Arabidopsis in variable light, which challenges the photosynthetic system, you can actually get an effect on fitness, which is dependent upon the amount of PSBS present in the leaf. However, there's never been a direct link to whole plant photosynthesis, so which is why we were doing this. Okay, so we were fortunate to have a collaboration with Syngenta, who made some transformants for us using the genomic sequences that were identified from the microarray experiment, and they generated overexpression and RNA interference lines for us. Okay, so we chose 3 genes: PSBS, beta-carotene hydroxylase, and PGR5, which regulates the cyclic, the rate of the cyclic electron flow around photosystem 1. So I'm going to talk largely about work that we've published already, and I'm going to mention some of the other work, but I'm a little bit limited in what I can say about it, unfortunately, until we publish it. So the PSBS transformants worked out very well. So this figure up here shows this is a color image, a falsely colored image of rice leaf segments measured for chlorophyll fluorescence, NPQ, and we see a several-fold variation in the amount of NPQ in these rice leaves, and this is related fairly closely to levels of PSBS through a Western blot. So we know we've changed the amount of PSBS in the leaf dramatically. If we measure photosynthesis, we find that the overexpressors and the wild type have approximately the same level of photosynthesis in terms of capacity, and the RNAi lines have a slightly reduced rate. This is supported by measurements of VC max from ACI curves, and we also see evidence of a sort of down regulation of Fe to Fm in the RNAi lines. But I'll jump straight to the interesting results, which is the relationship between NPQ and photosynthesis. Okay, and I plotted them against each other here. What we did here was we challenged the leaf by giving a train of light-dark cycles. Okay. Now the idea here is to continually induce photosynthesis by turning the light on and then off and then on and then off, etc. And because induction of photosynthesis takes a while, this challenges the photosynthetic system and increases the amount of photoprotection required. When we do this, we find surprisingly the overexpressors have a greater amount of photoprotection, yes, but they have a lower level of photosynthesis. And we also measured electron transport through photosystem 2, and this is reduced during induction in the overexpressors. So we think that there is a cost for overaccumulating PSBS in the leaf. So here we measure the amount of carbon assimilated during these so-called light flecks. Okay, so we use these results to create a kind of a schematic diagram of how we think protective NPQ should exist in the leaf. So perhaps the ideal productive state is obviously a high rate of CO2 assimilation but a low level of NPQ. If we accumulate a lot of PSBS, there's a risk that during fluctuating light we will have a reduced productivity, but if we lower the amount of PSBS to counteract that, we will have an increased risk of photoinhibition. So we think there's an optimization that lies somewhere between these two things, and the question then is what process dominates in crop canopies, and this is really what we're after. Is we can look at things in the lab, we can look at phenotypes, we can look at responses. The question is what processes dominate in crop canopies. We couldn't grow genetically modified plants outside, unfortunately, but luckily we had some glass houses at Nottingham that we just constructed more or less for this type of purpose. So these consist of large soil tanks, about 1.2 meters deep. We can control the water profile, but more importantly, we can grow crops with realistic canopies. This means the plants are interacting with each other as they grow, and we get what are called emergent properties of canopies. A plant growing on its own, as Xinguang Zhu said yesterday, a plant growing on its own does not behave the same as a plant growing within a community of plants. So this is what we're after. So this is a picture of my group carrying out a replicated randomized mini-plot experiment. And the light in this glasshouse is variable. It's supplemented by artificial light, but it only comes on when required. So if you look down here, well, first of all, you can see we get nice rice canopies that are large, productive, rates of biomass production are high. We get variable light in these glasshouses, which is important. And we measured, very briefly, we measured radiation use efficiency. This is what we do at the agronomy level. So we measure the amount of radiation intercepted using line sensors, and we also measured the amount of NPQ using monitoring fluorometers that we deploy continuously during growth, and then we measure biomass. As I said, I'm slightly restricted in what I can say, but what I can say is that we do see a higher radiation use efficiency, okay, before canopy closure, but not after. So we do this by plotting the accumulated biomass against the accumulated intercepted radiation, and it's higher in plants that have a higher level, a higher capacity for non-photochemical quenching. And this, to my knowledge, is the first time that anyone has ever shown this, either in a crop plant or Arabidopsis. So we think this is because during establishment and before canopy closure, there is a higher proportion of sunlit leaves in the canopy, which means the proportion of photoinhibition as a whole in terms of leaf area of the canopy is higher. As canopy closes and more leaves become shaded, we get an increase in canopy complexity, which means optimization of photoprotection and photosynthesis becomes much more important. Okay, so we also think from this that canopy architecture is absolutely critical, and we do need to integrate architecture with photosynthetic properties. Okay, I think I'm running out of time, so I'll just skip forward a little bit, excuse me. So that's really where we are with the transformation work. And we also started some mutant screening a few years ago, and this is ongoing work, so there's no conclusions I can come to. But we screened Halium's IR64 lines, not very many of them to begin with, although we are continuing this. And even just by screening a few hundred gamma lines, we find substantial variation in NPQ. We think it's a very sensitive component of photosynthesis. So, for example, here we have the wild type, and we have lines which show sometimes almost double NPQ and much lower NPQ. Okay? And we see variation in photosynthetic rate, but some of these are very close to the wild type. So that work is, so we think that's a very good source of variation for photoprotection. That's ongoing. I'm interested, we also did some screening as part of the C4 rice project. This was not for photoprotection, this was for compensation point. So we were looking for mechanisms which could give us which could separate plants which had a small change in CO2 compensation point. We did these in low CO2 chambers. So we grouped plants to leave 6 days in a screenhouse, transferred them to low CO2 and relatively high light for 6 days, and we measured the amount of chlorophyll lost. The reason for doing this was to get a high throughput. So with these chambers we got 1,700 plants screened in 1 week, which is really a lot. Okay. So this is what we did. We measured chlorophyll content with a SPAD meter before treatment, 30 ppm CO2. So ambient CO2 is 400. Okay, so this is well below the CO2 compensation point for a C3 plant. We measured the loss in chlorophyll. There's always a loss in chlorophyll. We selected for a low percentage chlorophyll loss, and we recovered putative candidates. And we found that very pleasingly, the amount of chlorophyll lost by the mutant population was actually lower than the percentage chlorophyll lost by the parental lines, which means that we can use it to screen for tolerance to high light and low CO2. Okay, so we're looking for lines down here. These yellow and green lines are C4 plants. So these lines over here actually overlap with C4 plants, which is very promising in terms of screening for CO2 compensation point. So we're just coming to the end of this analysis, and we're asking ourselves how robust is chlorophyll loss as a method for screening. We think it works well. So for a C3 plant, under these conditions, we'll have mostly oxygenase activity, high oxidative stress as well, and high carbohydrate consumption. And we do find in the candidates that we get a 5 to 10% lower compensation point in not all, but some of these high tolerant mutants. And this is largely caused by changes in, we think, in changes in respiration rate and carbohydrate consumption, but there's multiple mechanisms associated with this tolerance. So I think low CO2 is perhaps a little bit too harsh to select just for photooxidative stress. Okay, just to finish up quickly, the way forward. So from our transformation work, we have a proof of concept. We've shown that photoprotection has the capability of limiting biomass production. That's what we think. We've implicated canopy architecture and phenology, so we do need to phenotype canopy structure. I think this is something that's ongoing in many phenotyping programs, and also high-resolution canopy imaging. But the optimization at the leaf level is still not well understood at all. I noticed recently the first model has appeared for non-photochemical quenching in the literature just last month. So this is really ongoing. So we need to screen for novel genes and trait combinations. And the way to do this is to take, I think, is to take mutant populations and mapping populations. Again, phenotyping of canopy structure is going on, but we need to develop good screens under conditions of high radiance, maybe high temperature, and what can be called high excitation pressure. And I can't emphasize this enough. If we're going to screen for plants that are capable of doing well under high light and extreme conditions, we need to screen them under conditions of high excitation pressure. Okay? And this also must be informed at every step of the way by models that integrate canopy structure and dynamic photosynthesis, so the things such as Jingguang Zhu was talking about yesterday. Okay, just the last slide. So we recognise the importance of canopy architecture in optimising photoprotection, and we are currently working on techniques for imaging, for rapid imaging, and then slower high-resolution imaging of canopy architecture. We need to develop methods for rapid assessment using chlorophyll fluorescence, I think. And the main limitation for this is not the speed, but the way we can image chlorophyll fluorescence in 3 dimensions on a complex 3-dimensional canopy like rice. Okay, and I think field phenotyping is very important. Okay, so I think I will leave it there, and thank you very much for your attention.
Thank you very much, Dr. Erik. So, just a limitation of the time, only one question we accepted if you have. No question? Yeah.
So, Erik, in one of your slides you were showing in terms of light response in the canopy that a lot of the points that you measured were below the expected response, and were you thinking that's due to photo inhibition or some other? I was wondering if you have some leaves that are more in the shade in the canopy, if it might have less rubisco or some other reason that they would have a lower light saturate and be below the expected.
Yeah, okay, that's a good point. But those points, I should have pointed out, were from the upper half of the canopy, and we measured leaves that were in higher light. But because we were plotting it against light intensity, there was a variation in light intensity, and we had a paper where we looked at acclimation of light through acclimation of photosynthesis at different canopy positions, and by relating those, so acclimation would result in a lowering of rubisco content. So we can't rule out that some of those points are due to a lowered rubisco content, but we didn't measure a substantial reduction in rubisco content in the layers of the canopy we were measuring. But I can't rule out it did have an effect. Yes. Yeah.
Okay. Thank you.
So, thank you very much again for the presentation.